My team comprising of Jonathan Cox, Mass Pet Care, and Debbie Frost, a technologist here at Ag Research. We got funded from the Foundation for Research Science and Technology in the Technology for Business Growth stream. The funding was to enable us to develop a cat food that looks as natural as for humans. So basically, what is involved in this whole process is for us to take raw materials and extract from these raw materials the necessary matrix so that we are able to put that material back together and make it look like an intact whole tissue product. What we do is, after we remove all the unwanted materials like fat, connective tissue, is for us to be able to bring these three ingredients or even more ingredients together and then form them into a shape or size that we want, so that when we slice them, they will look as if they are one intact unit that has never been separated before. And for us to do that, we have to understand that each raw material that we are working with contain the necessary proteins in the right quantity. And for that to happen, we have to characterize the products.
To determine if our raw materials contain the necessary proteins and in the right amounts, we need to characterise these proteins. We do this by extracting the total proteins out of the meat sample, and then by separating these proteins based on their electrical charge or their molecular weight using either a 1 or 2D gel.
Once the proteins are extracted and then we run the gel, then we are able to determine from the gel whether we have the right protein in the right amount. And as you can see from these gels, we have a number of ingredients that we have run. And one of the ingredients, we have myosin and actin. These are the two major proteins that we are interested in because they are the ones that will form the glue. The other ingredients don't have the right amount. So what we'll do with this information is we'll now calculate and then have a composition or put together the right amount of these different ingredients so that we can have the right quantity of these cross-linkers proteins that will form the glue, so that when we hit the product, we'll be able to get a good bind.